Hello viewers, I am Dr. Vadiraj Tangor, Assistant Professor, Sri Jagadguru Tontadariya Government First Aid College, Mundargi. In this video is a lecture series on nanoscience and nanotechnology. In that, this is our third video. So this is for BSc program, Physics in paper 1. This is for 6th semester. This is third session for nanoscience and nanotechnology topic. In this video, I am concentrating on synthesis and characterization of nanoparticles. Learning objectives of this session is to know the concept of synthesis of nanoparticles, to understand characterization of nanoparticles, concept of surface plasmon resonance, concept of various techniques involved in characterization and synthesis of nanoparticles. Session outcomes: knowledge of synthesis of nanoparticles, understanding of steps involved in characterization of nanoparticles, idea about surface plasmon resonance, numerical idea about the size of the nanoparticles. Prerequisites for this discussion is concept about nanoparticle sizes, various laboratory techniques in characterization, idea about surface plasmon resonance phenomena. Methods of synthesis of nanoparticles. Nanoparticles can be synthesized by many methods. Most commonly used method is chemical reduction method, sputtering deposition method, electrolysis, thin film coating method called chemical vapor deposition method, pyrolysis, and in latest green nanotechnology. Many other methods like electric arc, molecular epitaxy method, sol gel, plant extracts. In this way, list is tailored to many methods. The most commonly used in all laboratory is synthesis of nanoparticles using chemical reduction method. We are taking one example, synthesis of gold nanoparticles by chemical reduction method. Method is very simple. The things required for this method is gold auric chloride to get the nano gold particles, and reducing agent is sodium citrate, and in some laboratories they are using sodium borohydrate as a reducing and capping agent also, and magnetic stirrers to continuously stirring with a hot plate. These are the instruments that are required for our chemical reduction method. What we should do is, first step is bring to boil 50 ml of 2.5 into 10 to the power of minus 4 molar chloroauric acid solution. Keep it in a magnetic hot plate with a stirrer. Then add 0.16 to 1 ml of 34 millimolar sodium citrate solution to the boiling solution while stirring. This sodium citrate is a best reducing as well as capping agent. Reducing means in a chloroauric acid solution, it removes all acid part, chloride part as a reduce and only keeps gold particles. Because of continuous stirring gold particles, it reduces the sizes of nano size. The variation of sizes of nanoparticles can be tailored by quality and quantity of adding the sodium citrate in a numerically calculated manner. Then continuously stirring will be takes place. After a minute will be faint blue, then darkening over 5 minutes to become a brilliant red. The size of gold nanoparticles can be controlled by varying the amount of sodium citrate solution. The above procedure can be controlled size of the nanoparticles. In this way, we have reduced the gold nanoparticles, but gold nanoparticles in that scale, in the nanometer scale, they are hyperactive. Therefore, we should make it capping to reduce the activity. Better is certain stabilizing agents like a phytochemical matrix we are using, like a thpal and proteins, some things we are coating for the gold nanoparticles. As a result, these nanoparticles can be withstand for a month together. Otherwise, because of hyperactivity, it will be lose its stability. Within an hour, fraction of minutes, it will becomes a common gold. Therefore, to enhance the stability for a longer period, we should do the capping for the gold nanoparticles. So, gum arabic is a non-toxic food additive. It may also be act as an capping agent. Complex structure of gum arabic responsible for the stability of the gold nanoparticles. Make colloidal gold from gold chloride. Making colloidal gold by low voltage using electrolysis method and citrate synthesis. These are the many methods for the synthesis of nanoparticles. Now, whether to check synthesized gold nanoparticles are nanoparticles or not can be easily checked by taking the solution in a test tube and put it for an examination in an ultraviolet visible spectrophotometer. A beautiful surface plasmon band is observing nearly about 520, 530, 540 nanometer. This represents we have synthesized the gold nanoparticles. And another chemical method, whether we have synthesized it properly or not, can be easily checked by destroying his nature by just adding sodium salt NaCl. Then suddenly surface plasmon resonance will be shifts to around 700 and the peak will be broadened. This represents that whatever we have synthesized the nanoparticle is accurate, and the peak position of 520 nanometer, 540 nanometer, it's all characteristic of particular size of the nanoparticle. Therefore, size of the nanoparticle can also be calculated by placement of the surface plasmon resonance P. Now, another method is compatible methods are take a gold auric chloride, inject thpal in water, stir for few minutes, and the solution becomes pink color. This is around about 16 to 20 nanometer sizes of the nanoparticle. The TEM image, SEM image, scanning electron microscopic, tunneling electron microscope images, they will express the size of the nanoparticle. And because of the reduction method is it is not an eco-friendly. Therefore, we can synthesize of green gold nanoparticles by using a green nanotechnology is adopted towards the fabrication of biocompatible gold nanoparticles and in a non-toxic way produced by reduction of sodium auric chloride by trimeric alanine based phosphine in agaricus water. So hence we can synthesize around about 4 to 12 nanometer and capped by some glycoproteins, and it can withstand for 6 months together, and it is eco-friendly method. And in a reduction we are observing various chemical wastage hazardous that hazardness cannot be observed in case of green method. The gold nanoparticles can be capping by gum arabic by shown like a picture, some image, tem image, everything can be seen, and stability of the gold nanoparticle is also extended for a few months together. The tunneling electron microscope image, scanning electron microscope image, it explains the size of the nanoparticles, micrometers, nanometers in this way. The formation of gold nanoparticles can only be confirmed by surface plasmon resonance. Surface plasmon resonance for gold nanoparticle is around about 540 to 550 nanometer, and at the peak is around about 545 nanometer. It confirms that size is around about 4 to 16 nanometer sizes of the gold nanoparticle. So we know surface plasmon resonance is whenever incident wavelength of the light is matching with the size of the nanoparticles. As a result, sudden bump is observed in a ultraviolet visible spectrophotometry, and that peak is called surface plasmon resonance. And the green nanotechnology has been started by our Karnataka Indian great scientist. Now he is working in a Missouri American University. He is treating the cancer therapy using green nanotechnology. He is called Katesh Katti, and for his work, our Karnataka University has been honored with a doctoral degree. And he has synthesized gold nanoparticle by using cinnamons, cumin seeds, pomegranates, and turmeric. In this way, by using greenery objects, he has been reduced the gold salt into gold nanoparticles. So such a beautiful person, he is working on cancer therapy by using nanotechnology. Hence, he is most interested in a green nanotechnology. Nowadays, he is called them as father for green nano. And he has also observed synthesis of nanoparticle by using soybeans. Why is nanoscience is popular now? So science is popular because of these instruments to see the nanoparticles. One is called SEM, TEM, STM, AFM. You should remember the analogy of these terms. SEM is scanning electron microscopy, TEM is transmission electron microscopy, STM scanning tunneling microscope, AFM atomic force microscope. With the help of these instruments, we can explain the characteristics of the nanoparticles. So, in a scanning tunneling microscopy, because of its moment on the nanoparticles, it can measure the size of the nanoparticle. The tip is moves on the surface of the nanoparticle. As a result, it measures the size of the nanoparticle. So STM, it moves, and here it forms the clouds, and these clouds can be measured with help of the scanning tunneling microscopy. As a result, we can measure exact size of the nanoparticles. So for the first time, IBM peoples, they have been seen the atoms using these kind of instrumentation, and we can see the IBM has been measured by has been observed keeping the atom one by one. Such a huge microscopy development, we can see the nanoparticles, we can see the atoms nowadays. So because of keeping the atoms, it can produce the waves. Those waves can be studied by using Schrodinger's wave equation. This is all my today's topic. So I will be close my topic. So thank you one and all for your such a cooperation with this video in seeing up to the end. Thank you one and all.